concept, what are we going to learn? If this is 3' to, can you see the board, right? 3' to 5'. And this one is 5' to 3'. Okay. Now, when you learn about the transcription unit, so what is this transcription unit? Okay, it consists of promoter. We call that to be promoter. See, I'm drawing it here. This is the structure. Okay, we call it as promoter. What is this promoter? And towards this end, you have a structure. You call this to be terminator. Terminator. Okay. So first one is promoter. Okay. Next one is terminator. Now, what is present in between the promoter and the terminator? They are in between. You call this as structural genes. What are these called as? Structural genes. Okay. So this transcription unit consists of promoter, structural genes, and terminator. These are the main three parts present in this transcription unit. Okay. Template is something what is needed to produce the another copy of it, right? Okay. So here 3' to 5' act as a template strand. The message that is present on this template, it is going to be copied here. What is that? I'm going to draw a line here. An mRNA which is being formed with the help of this template strand. Template strand helps in forming the mRNA. So how this mRNA will be developed? The message that is present in the template, that is the DNA strand, will be copied to the mRNA strand. Now, what is the difference between DNA and mRNA is, mRNA normally we have studied about, it is single stranded. So 5' to 3'. This mRNA always polarity is 5' to 3'. That is very clear, is it not? It is a single stranded molecule, I told you, right? Any doubt with that? Okay. If this strand acts as a template, it will be parallel. That is why 3' to 5' act as a template. Have you understood now? Now this information, it is being copied to the RNA, mRNA. How is that copied? With the help of 3 steps. First step, you call it as initiation. What do you call it as? Initiation. First step, what is involved in transcription is initiation. What is initiation? Starting. Something which initiates the process. Something which helps in starting the process of transcription. What is that? Okay, RNA polymerase is involved. So what is the function of this RNA polymerase? This enzyme, when it comes and binds with the promoter, this RNA polymerase, when it binds with the promoter, okay, here you have a factor which is called as sigma factor. You know this symbol, right? Sigma. Okay, what do you call this to be? Sigma factor. So this sigma factor helps in binding the RNA polymerase enzyme to the promoter. For what? Initiating the process of transcription. Any doubt? When RNA polymerase, when it comes and binds with the promoter, with the help of the sigma factor, the initiation will start. Okay, right? Okay. Then what is the next step? Second step is elongation. What is elongation? This RNA polymerase, after initiating the process, it continues the message, that is the genetic information that is present in between the promoter and the terminator. It has to be copied to the mRNA. That is nothing but you call it as what? Structural genes. So after initiation, elongation. What is it? Elongation? The process continues. It elongates the mRNA, which is formed, it elongates. What is that elongation? All the messages, genetic information present in the template strand, it is copied to the mRNA strand. Have you understood? This process, you call it as elongation. So how long till it continues? We have studied that DNA is 2.2 meters. Do you remember? In DNA packaging, right? So how long it continues? There will be a break where there is a terminator. Here, you use a factor which is called as rho factor. You would have learned about the term rho factor. Do you remember? Rho factor. So, rho factor helps in stopping the process by the name terminator. Terminates. Terminates meaning stopping. It helps in stopping the process, which helps. Okay, right. So here, when this RNA polymerase, when it elongates, when it finds the terminator, okay, when the RNA polymerase binds to the terminator with the help of the rho factor, the process will be stopped here. Can you understand? Any doubt here? What did I tell you? Initiation when the RNA polymerase enzyme, when it binds with the promoter with the help of the sigma factor, the process will start. That is called as initiation. So that is the first process of transcription, right? Okay. So after the process starts, it has to elongate the process. The information that is present in the template strand, it has to be copied to the mRNA. Okay. So elongation. Then after that, next step is termination. Termination is the last step of this process. Okay, so what happens during termination? The transcription will get stopped here with the help of the rho factor and the terminator. Have you understood? Okay, so the information that is being present here in between the promoter and the terminator will be copied to the mRNA. So what is that information? This is DNA. Okay, only for A you will change it as U. Now guanine, cytosine. Cytosine, guanine. Adenine again, uracil. Thymine again, adenine. Guanine, cytosine, guanine, adenine, thymine, adenine, guanine, cytosine, guanine. Are you clear? Okay. Thymine pairs with adenine only. Don't get confused. This is very important 3 mark or 2 mark question in your board. Ask you to write mRNA, or they'll give you mRNA and they'll ask you to write the template strand. Anyway, you're trying to understand what is that concept. That is very important. Okay. I'll give you some other, what is that? Question. This is the coding strand I'm giving you. Okay. Adenine, thymine, guanine, adenine, thymine, guanine. Okay, right. Now, when you learn about this promoter, I forgot to tell this thing, promoter, okay, this promoter basis always starts with TA, it goes on like this. Promoter nucleotide basis always starts with like this, TA. So normally, this promoter box, you call it as Tata box. What do you call it as? Tata box. TA. The basis will start from TA. So in short, you call this promoter box to be Tata box. So only with the help of this promoter, the RNA polymerase can bind to the promoter with the help of the sigma factor to start the transcription. That starting step, you call it as what? Initiation. After that, the bases that are present in between the promoter and the terminator, you call that to be structural genes. These are going to be copied to the mRNA. This process, you call it as what? Elongation. After elongation, there is a terminator over here. When the RNA polymerase binds to the terminator, with the help of the rho factor, it gets stopped here. You call it as termination. So initiation, elongation, and termination. Sometimes the rho factor also helps in elongation process also. Okay, so these are the 3 steps involved. Now, once it gets terminated, this mRNA comes out. Where does this take place? It takes place inside the nucleus. This is the cell. This is the nucleus. Can you understand? DNA is present inside the nucleus. We are talking about the nucleus that is present in the DNA. DNA is present even in mitochondria also. We are learning here what is present in the nucleus. Can you understand? Okay. After the transcription, the DNA has to come out from the nucleus. Where did you study transcription? After that, mRNA is formed. This mRNA will be useful for the synthesis of proteins. So where is this protein synthesis take place? Which is, and that ribosomes are present in the cytoplasm. This is the cytoplasm, is it not? So ribosomes are present in the cytoplasm. This occurs where? Inside the nucleus. Transcription takes place inside the nucleus. Now that mRNA, what is being synthesized, formed, okay, copied, that has to come out from the nucleus. Can you understand? When it comes to the ribosome, next process can take place. Now there is a question for you. We have learned about the eukaryote and a prokaryote. Eukaryote, I told you, there is a well-defined nucleus, whereas prokaryote does not have well-defined nucleus. Right? Correct? So here, this is a well-defined nucleus, where the nucleus has the nuclear membrane, and here you have the double-wall membrane, all those things. Plants and animal cells, this type take place. Right? In prokaryote, there is no well-defined nucleus. Then from where will that comes out? mRNA after the formation. Okay, that is what we are going to study. So this is general transcription, what happens. Okay, all these you have understood, right?
If you like this video, share and subscribe my YouTube channel and follow me on Facebook and Instagram. Thank you.